Let's welcome Caleb.
Hey, got it. We're recording. Thank you so much. Thank you for inviting me over today. It's a pleasure to be here. I'm particularly appreciative that you're here on a Friday afternoon. I mean, I feel my energy levels flagging. So it's great to have some people to talk to. Yeah, so let me plunge in. Do we keep the lights on, or it doesn't? I'm sure it's fine either way.
Intermediate.
Whatever people prefer is fine with me.
You want to open the windows a little so that energy level doesn't flag too much?
It's all good. So, so I was thinking about what to talk to you about today. I'm going to talk to you about something a little different. It may come across as completely wacky and crazy, and that's fine. You know, I'm not afraid of that. In my work, as Natalie indicated, I've jumped around between different areas. So really what I'm going to talk to you about is an idea and how that relates to our quest to understand where we can look in the universe to find evidence of living systems, but it also digs much deeper into really thinking about what we mean when we ask the question of, can we find other living systems in the universe? So let me plunge in. I've used this title, Rebuilding the Habitable Zone, bottom up. So in astrobiology, the habitable zone is a thing that a lot of people spend time worrying about. And if you ever talk to an astrobiologist, chances are this term habitability will come up. So the idea of habitability has really been a reference point for astrobiology for a very long time. And it's, you know, I could be cruel and say it's a way to say something about the likelihood of life, even if we know nothing. And in many ways, that's what it is. It's a way to narrow down which exoplanets we might prefer to look at if we're trying to look for biosignatures, which places in our own solar system we might, you know, focus our attention on, because there are aspects of the qualities of those places that we think lend themselves more to the existence of life. So many of you in the room will know all of this already, but I'm just going to go through it very briefly just to review it a little bit. But this idea of habitability, of the likelihood or potential for life in a particular environment, isn't just about, you know, deciding where you're going to point your $10 billion telescope like JWST or do other things. It's actually an important factor to include if you are studying putative biosignatures, signatures of chemical abundances in a planetary atmosphere, maybe some peculiar chemistry on the surface of a world like Mars, or on the surface of Titan, or anywhere in our own solar system. You know, it's a useful tool for evaluating false positives. I suppose you find a biosignature. Let's say you find evidence of an oxygenated atmosphere around a rocky planet elsewhere in our galaxy. You know, oxygen is actually a very tricky biosignature, but that's another story. But if you find that positive biosignature in an environment that doesn't meet your standard criteria for habitability, and I'll give you a specific example in a moment, that's an important thing to know. It's a potential way to eliminate false positives. The habitability isn't just a way of categorizing things. It's an important part of the toolkit that we have in our search for life in the universe. So in exoplanetary science, the most commonly talked about kind of measure of habitability or habitable zone is this thing called the circumstellar habitable zone. Again, many of you are going to be very familiar with this. And this is really just a first pass way to ask where a planet orbits its star, if that's in a range where there's the possibility of liquid water on the surface of the planet. And the interesting thing, just a tiny bit of history here, is this general idea that water is super important for the existence of life goes back a long way. Newton kind of understood this. He said, our water, if the Earth were located in the orbit of Saturn, would be frozen. If in the orbit of Mercury, it would depart at once into vapors. That's pretty amazing to say that in the 1600s. And it really foretold this idea of a circumstellar habitable zone. You need to be the right physical location around a star in order to, based on a variety of factors, possibility of sustaining liquid water on the surface. And even, around the early 1900s, people started to recognize with a little more focus the importance of water, especially in the liquid state. So this is Edward Maunder. And Maunder is known for his study of a certain period in the history of our sun, where there's a Maunder minimum of solar activity, which I keep forgetting when exactly that was, but I assume it was sometime in the 1900s. But he also was interested in, I'm sorry?
It's the time of Louis XIV.
That's it. Yes, thank you.
Or European Little Ice Age.
Yes, thank you. Excellent. So Maunder was also interested in circumstellar habitability. He didn't use that terminology, and he wrote, circulation of matter, which we call life, the metabolism of vital processes, requires for its continuance the presence of one indispensable factor, water. And then he says the search for life on Mars is essentially a search for water. Now, I'm not quite sure how he got to that, and obviously people were thinking about Mars in a rather different way in the 1900s than they are today. But you could almost lift that and put that in a piece of NASA paperwork for motivation for a lot of Mars exploration in the last couple of decades. So people have been recognizing this as an important factor for habitability. So let me just say this more explicitly. Today, we understand that water is this critical solvent, reactant, product, and catalyst. It does all of those things for biochemistry. It's absolutely vital to biochemistry on the Earth as we know it. And another interesting aspect of the presence of liquid water, which I think gets overlooked quite a bit, especially by people like myself who come out of astrophysics and whose biology is still a work in progress, is that the same conditions that allow for liquid water and biochemical reactions, in other words, conditions of temperature and pressure, also ensure thermal motion at the molecular level that actually helps drive important biomolecule function. And this is something really fascinating that I will show with a cartoon. So this is not a molecule. This is just a graphic to illustrate the principle. But it turns out that for certain biomolecular machinery, things like RNA polymerases, ribosomes, these are complex molecules containing hundreds of atoms, if not thousands of atoms. They can actually harvest the thermal motion around them. And that harvesting of thermal motion drives important biomolecular function. It literally helps the molecules move around, ratchet themselves into new positions to pull energy carrying ATP molecules or whatever amino acids out of the solution. So this little cartoon is just an illustration of what I mean by Brownian ratchet. So here we have these veins, these green veins, and they're being bombarded by the thermal motion of those red particles, the atoms or molecules or whatever. And just by chance, every so often, the bombardment, the forces acting on those green veins pushes the system just far enough in one direction that the ratchet can move along another step, another tooth in the ratchet. It can't go back again. So in this way, you can actually extract useful work from random thermal energy in the environment. It's pretty remarkable that life does this. Life taps into that for some things. And it's, again, the same sort of general conditions that allow for liquid water also allow for that level of Brownian ratchet energy harvesting. Water in all its phases is critically important for climate states. It's important for hydrological cycles, obviously, the cycling of other things that are dissolved in water. And it's even important for things like plate tectonics. Presence of large oceans on the Earth plays a role in the precise mechanics and placement of plate tectonics, which are in turn important for the overall geochemical, geophysical cycling of the planet. So water is important. And circumstellar habitable zone is a good idea, but it's not the only thing that defines habitability. We know that habitability must be contingent on a whole bunch of other things. I'll just list a few here. So it has to be contingent on the availability of certain key elements. I mean, things like carbon, hydrogen, oxygen, nitrogen, yeah, they're everywhere. right? But life also makes use of certain elements that are much lower in abundance, both cosmically and on the surface of the Earth. So things like phosphorus, for example. Phosphorus plays an important role in the phosphate backbone. Things like DNA. It also plays a role in energy-carrying molecules in cells, things like adenosine triphosphate. Phosphate is incorporated there. And then things like molybdenum, which is kind of not necessarily what many of us think of as important for life, but molybdenum plays a role in nitrogenase. Nitrogenase enzymes are a central part of fixation of nitrogen out of an environment and the nitrogen cycle. So habitability for life as we know it hinges on some of these things too. It absolutely hinges on the availability of free energy. This is something I'm going to come back to in quite a bit more detail. So free energy is energy that can drive metabolism. It's closely related to geophysical cycling because you need fresh, raw chemistry constantly coming into availability for living things. And we understand that you have to have really an excess of free energy, chemical energy. It could be energy derived from stellar photons, and I'll come back to that in more detail as well. It seems that you need an excess above the actual biological needs for a system to not be struggling, for a living system or biosphere to be functioning well. And then there are other things, many other things going into habitability. Planetary geophysics I've mentioned, but contingency, the history that precedes life at a certain point on a planet is probably extremely important for setting the habitability at that moment. And a good example is the oxygenation of Earth's own atmosphere, which is, of course, a result of living things, but it's made the planet more habitable, right? Because it's opened up a whole array of biochemical mechanisms that didn't really exist when oxygen was not so freely available. So what does this mean? Well, it means habitability is a pain in the ass to deal with because there are so many things that you really need to be considering, and it's hard to weight one thing above another. Liquid water, absolutely, but even liquid water, you know, there are potentially circumstances where you don't need globally available reservoirs of liquid water. It could be very, very localized. So this has had me thinking about ways in which we might try to somehow pull these threads together and to also at the same time get closer to thinking about what it is we're really looking for when we're looking for living systems. You know, what is life? And because that's a question that is a slippery question. It's hard to answer. You ask, there's an old joke in astrobiology, you ask 100 astrobiologists, give me a definition of life, and you'll get 100 different answers. So is there a way to perhaps do a slightly better job? So that's the question I'm going to try to unpack a little bit and give you my suggestion or our suggestion for this, which may sound a little wacky, but if you bear with me, I'll try to explain. So is there a way to rebuild fundamental definition of habitability that does more to unite all of those phenomena and also somehow tap into some fundamental principles that go a little bit deeper than just, well, you need a molecule that does this or you need a molecule that does that. So what I'm going to say, I'm going to jump straight to the sort of the main line here, which is a proposal that living systems are really about computation. Now, when I've given this talk, and I've only given this talk a couple of times before, often when I say this, the biologists in the room roll their eyes and go, oh my God, here we go, a physicist trying to do this. Computer scientists get really interested, but also confused. And the rest of us wonder what I drank over lunch or this morning. I haven't touched the wine yet in the room, so, but I'm looking forward to some of that. So all I'll say is don't panic, okay? Because when I talk about computation in this context, I'm talking about something a little bit different than the way you probably usually think about computation. So let me dig into that. So in many ways, I'm talking about computation in very general form. So computation as a set of physical processes that act on information that is represented in states of matter. So for example, molecular configurations or electron energy levels, whatever you want. Matter can represent information, and that information and those states of matter can be manipulated to produce something else, and that is essentially computational processes. Now, what's nice about that is it ties directly to the sort of emerging picture of what life is that we've actually had since the first ideas of genetic inheritance, our decoding of the molecular basis for genetic inheritance in DNA and RNA and so on, that living systems really do seem to be about information that can both exert itself over matter and be modified by matter. And living systems are about physical processes that utilize information in the living system and mutual information about the environment. So some of this is terminology. Mutual information has a very specific meaning in information theory. It's literally the mapping of information from here into here, so that two things in the world contain a map of the same information. That's all that mutual information means. And as I go through this, hopefully some of this will make more sense if it's not already making sense. So, we understand that in things like DNA and RNA, we talk about the genetic code. That's an oversimplification, but it does relate to this idea of information propagating itself in the world, influencing matter, exerting itself over matter, and so on. So to talk about life in terms of computation is perhaps not so crazy at all. And there's been a considerable body of work. Don't know why I put myself here. There were many other people who've done this. David Krakauer, for example, the Santa Fe Institute has written some beautiful papers about really the physics of self-propagation and the relationship to information and prediction-based action. So organisms' life also does this thing of predicting the future. Right? Even a bacterium, if it senses a chemical gradient and follows that gradient, it's essentially taking information about the environment, turning it into mutual information because it's somehow encoding it in itself, and then making a prediction that if it follows that chemical gradient, something will happen that is presumably a positive thing that will enhance its capacity to survive in the future. Okay. All of which is really a capacity that we can label as computation. And the nice thing about this, the appealing thing about this, is it's more fundamental and it encompasses biology, and I would claim also biology's extended phenotypes. So everything in this room is an extended phenotype of us. The table, this meeting owl, my computer, the screen, the lights, right? It's all stuff that has come from us, right? Our manipulation of matter, but it's external to us. Right? It won't move through time with us in the way that our genes move through time with us, but it is part of our existence, extended phenotype. Our bodies are extended phenotype, even within our cells are extended phenotypes, the expression of our genes on the world. And computation happens in all those levels, cells to organisms to all the stuff in this room to technology, I would claim. So there's something appealing here about this idea of computation and sort of universality of it. I mean, as I keep going, hopefully some of this will unpack a little bit more. So again, I'm cutting through some more complicated arguments just to give you the idea here, the essence of this. So what if, instead of asking about habitable zones in the traditional way, we ask about computational zones in the universe, places where computation, as I've defined it in this very general form, can take place. But that also seems kind of what we're looking for. We're looking for places where information can exert itself on matter, can propagate into the future, undergo selection and evolution, and so on and so on. And when you start thinking this way, looking through this lens, what's nice is it feels like it begins to pull together those various threads that I mentioned earlier in traditional ways of thinking about habitability. Pulls together planetary information about planetary conditions, pulls together biological information, ecological insights, and so on. And the idea here is much like with the traditional circumstellar habitable zone, there have to be constraints. So the circumstellar habitable zone, the constraint is you have to have liquid water, right? So it winds up mapping essentially to a constraint in temperature, and that maps to a constraint in orbital range for rocky planets and so on and so on. Here, I claim we have three types of constraint on computation. There's capacity, energy, and instantiation. Let me just expand on that a little bit. So capacity is literally just the number of available states for carrying information. So it could be the number of molecules, it could be the number of bits, digital bits, it could be other structures. Doesn't really matter. It's just, how much stuff can you emplace information? It could be every atom composing the Earth, right? Or it could just be every atom composing the outer surface of the Earth, or some subset of that. It's very, very general. So capacity is about, the number of states available. It's not really talking about memory per se, but you could look at it that way, like computer memory. But it's also to do with the maximum possible rate at which those states can be manipulated. And there are going to be constraints on that from physics. How quickly an electron can change its energy level in an atom, how quickly a molecular bond can form or break, and so on. And then we have energy. If you're going to carry out computation, if you're going to exert change in the world, you need energy. You need useful work, availability of useful work, or energy for useful work. And that's interesting because it's also kind of agnostic. It could be fast or slow changes. It doesn't really matter in this case. It's just a general constraint on energy. I'll come back to that in rather more detail in a moment. And then what I call instantiation or substrate is what your computing process is built out of. Okay, so and it's a sort of a kind of a computer science term, like instantiation. How do you instantiate something as a piece of code? But this is really just ways that computation may be carried out. That could include information about the elemental composition, the molecular structures that you're using, but it can also include information about the environment and things like temperature. So in principle, these three constraints let you fold in pretty much all the things I listed in that more traditional list of habitability. The trick is to figure out how to do that, okay? So I often say some of these ideas that I get excited about, it's like throwing a rope across a chasm and then dancing across it because you have no idea if you're going to get to the other side or not. So that's kind of where this work is at the moment, just so that you understand if it's not already very obvious. So let me talk a little bit about the constraint of energy for computation, especially as it refers to biological computation. That's an interesting inroad to this. And I think it gives the first example of why this may be onto something, why this may work. So the interesting thing, and this is summarizing many, many papers, a long history of people worrying about this, that we know already from the field of information theory and from thermodynamics that there is a natural hard lower limit on the energy use when there are informational changes in the world. So for example, just to put it in recognizable language, if you have an irreversible bit operation, I erase a bit of information. It could be written in a piece of silicon, it could be written in a molecule, it doesn't matter. It turns out that from an argument based actually on Boltzmann's equation, entropy being Boltzmann's constant, natural logarithm of the probability of a given microstate, you can derive what's called the von Neumann-Landau limit. And that says there's this energy here, E Landau is the Boltzmann constant times the temperature times the natural log of 2, because this is a binary operation. You erase a bit of information. It turns out that there is this universal limit on how much or how little energy will ever be involved in that process. It could be dissipated energy, it could be energy required to make that happen. And it goes to some quite deep physics, and you can go and use Wikipedia and dig through this. It's really interesting. And what's particularly appealing about this, the only thing this really depends on is the environmental temperature. The warmer things are, the more energy is required to perform an irreversible bit operation. That could also be the merger of two computational streams and so on. It's a kind of a general catch-all. And this is part of what got me interested in this, because it's like, oh, I can now plug in an environmental temperature and get an estimate of the minimum amount of energy anything, including biology, is going to have to use to perform an operation that's part of computation, as part of the whole process of living systems. It's very appealing. And some other people have also kind of cottoned on to this, including Chris Kempes, also at the Santa Fe Institute. Chris is a fantastic, if you want a good talk, get Chris to come and give a talk. It's a great talk. So people have kind of thought about this and looked at things, just to give you an example, like the computational efficiency of RNA translation. Okay, so that's where you've got a strand of RNA in a cell, and you have a ribosome running along it, and what it's doing, it's pulling amino acids out of solution and linking them together to make a polymer, to make a protein. That pulling of an amino acid, connecting it onto a chain, can be seen as a computational process. It can be seen like a bit operation. And it turns out that if you do the maths and you look at, you have to incorporate things like the probability of drawing particular amino acids, what the various concentration of different amino acids is, and so on. But you can do it. I've skipped that here. You end up with something that scales with the Landau limit. In fact, an RNA translation, the least energy you can possibly use to do that anywhere in the universe is about 10 times the Landau limit. Landau limit is temperature dependent, so roughly room temperature, that's about 10 to the minus 19 joules. Okay, for a single RNA translation event. And this has been sort of verified. You can also, as biochemists, go and look at ATP, the energy carrying molecule, adenosine triphosphate. It takes about 4 of those. Actually, it takes exactly 4 of those to form a single amino acid attachment to a growing polymer in RNA translation. And that turns out to be really close to this value as well. So it all kind of makes sense. There's a degree of experimental validation that this is true. So what is so nice about this is we now have this absolute universal lower limit on how much energy a living system is going to have to exert for this kind of process, right? And so we can take a jump and say, well, let's talk about biological operations per second, or BOPs. I thought about, should it be BOPs or LOPs, living operation? I don't know. I don't know what the best acronym is. BOPs is okay. Biological operations per second. So this is going to let us map some of those constraints of capacity, energy, and substrate together. It could be applied to Mars, the interior of icy moons, the entire solar system, exoplanets, whatever. So very, very simply, we assume some kind of efficiency of energy use, and I'll come back to that in a slightly different context in a moment, in a sort of worked example. We can obviously just write down an upper limit for some biological process X. The number of biological operations per second is just how many computations are happening per unit time. And we can write that as the energetic cost per computation times the available energy that you have to do that. So some efficiency factor times total energy over time, or the power flowing through the system. It could be chemical energy, available energy. It could be stellar photons, and that's the example I'll show you in a moment. Whatever. This is very, very general. So we can come up with this absolute upper limit. So how many biological operations per second be happening for a particular process, which is in terms of over some number times the Landau limit. So this number A sub x is like the factor 10 I had in the previous slide. It's just different biological processes seem to require different multiples of the Landau limit to be performed. And this is something we can experimentally determine. So just 1 over that times this efficiency times the power. Okay. And one could go slightly further with this and say, well, that's what you could do if all the energy available for biological operations could be accessed. That's probably not going to be the case. You may not have enough molecules You may not have enough amino acids to do this, to reach this maximum potential number of biological operations. So you need to add another factor in, so you can put in this fraction here, fraction of maximum possible operations, actually instantiated. I'm not going to go too deep into that. Just a convenient way of saying what you're actually going to get in a real living system. It could be an organism, it could be an entire planet. It's going to be whatever the upper limit is, always going to be that limit defined by the Landau limit, which is a fundamental piece of physics, times some fraction that says, you know, how many instances on this planet are actually capable of doing that? How many amino acids do you have? How many strands of RNA at any given moment are undergoing translation and so on? Something you don't know ahead of time, something you're going to have to figure out and determine. And I'll try to give you an example of that. You know, very briefly, once you start down this road, you can start to do things like say, well, an entire biosphere is going to be some summation of individual biological computational processes. What those are, we don't quite know yet. There's definitely going to be things like RNA translation or transcription, other things that are fundamentally informational in nature. And the interesting thing about that is we don't know quite what the weighting is for each of these, but there's also a potential here to begin to construct a map of computational hierarchy for living systems, which is, you know, something we could probably start to do already. It's a very different way of looking at just conventional biology. This is why I say some biologists rolling their eyes at when I'm talking about this, because it's like, yeah, well, we sort of know this already. Right, but putting it together in this framework is a little bit different. Eventually, perhaps you can even ask why certain computational processes in a living system are preferred or more energy hungry than others, and that must relate to deep questions about evolution. But that's going off on a tangent. So let me come back to showing you a couple of quick examples, just to give you a sense of how you begin to apply this. So the first thing is, okay, I've thrown up that expression for an upper limit to the number of biological operations, let's say on a planet. Am I talking about 10? Am I talking about 10 trillion? What kind of range are we talking about? Well, we can actually make some kind of estimate of this. So earlier works have looked at a version of this and actually tried to understand through a sort of argument of just taking an inventory of how much extant DNA and RNA we think is on the planet, on the Earth, and just sort of plugging the numbers together. And there's been an estimate made that the Earth's total transcription rate, or the number of nucleotide operations per second, again, that's, sorry, I've switched to transcription. So this is when we're reading out a piece of DNA and building a strand of RNA in a cell. This has been estimated at being about 10 to the 39 per second globally for the Earth's entire biosphere. Well, okay, does that make sense? Well, the interesting thing is, if you start to put that into the formalism I just showed you, you can put in what you think the minimum amount of energy is going to be needed for this. Say the typical, you know, temperature on the surface of the Earth, you plug that into this Landau limit expression, you put it into the previous equation. What it says is if this were true, then nucleotide operations per second on the Earth would be gobbling up 10 to the 20 watts of power. There's a bit of a problem with that because that's 1,000 times the total solar power absorbed at the surface of the Earth. So no, it can't be that. So that's important to understand. But then can we actually get a better estimate of this? Well, I tooled around with this and just, again, cutting some corners just to show you, you can kind of go back to what we know about the power use of individual microbial cells. There's been a lot of work done on this for various environments, various extremes, things like methanogenic organisms and things living at sort of slow lives in the deep silty bottoms of our oceans and so on. But you can see that there's a range in known microbial power use per cell. So that's incorporating whatever the cell is doing, right? All computational processes, all other metabolic processes, and so on. But this is kind of a measure, or it gives us a way to estimate that capacity that I talked about. Because the essence of this, if you play with the numbers, to make the power budget of individual microbial cells make sense, given that we know what the average gene length or average genome length is in those cells, we can actually come up with this estimate of the fraction of genome that is undergoing transcription at any given time globally. I'm not sure if that's going to make too much sense to you, but the bottom line is we can make a correction to that previous estimate of total energy use or total power use, and make a correction to this global box nucleotide, biological operations per second nucleotide. So it dropped by several orders of magnitude. It was 10 to the 39. To make sense energetically, it's probably no more than 10 to the 30 to 10 to the 32 per second. What that means is the power budget for the planet given over to these transcription events, assuming most of the planet is microbial, which is actually a fair approximation, physicist approximation within a few orders of magnitude, says that the power budget for that is around 100 gigawatts to 10 terawatts, which is kind of nice because that fits right inside of the roughly 100 terawatt limit that we generally consider for the overall biosphere in terms of its biochemical energy use. You had a question. Yeah.
Trying to remember the number you had for the 10 times Landau for minus 19 joules.
Oh, yeah.
10 to minus 19 joules. So I should read from that the typical bacteria is kind of doing a bit per second of transcription-y things.
I think that's about right.
Give or take,
Yeah,
but these
yeah,
energy for other things
yeah,
too. So that
yeah.
that would be another framework to think of it. But where did can you go back to the previous slide?
Yeah.
Just to trace the numbers.
Yeah. Okay.
So, right, is that saying that estimate of nucleotide operations per second is just extremely wrong?
Yeah.
I mean, it is basically,
Okay.
yeah. Yeah. And the thing about that estimate, it paid no attention to the energy required for that. It was simply based on estimates, experimental estimates of the rate of transcription happening in cells.
But those cells must therefore be using more than that 10 to the
Somewhere,
minus 18 or
yeah,
those cells must be really perky cells compared to the typical
exactly.
bacteria.
So either that was the case, or either there's some. I think it's either that they were looking at, the most active human cells possible, or stem cells or something like that, or there's just some experimental error there, a significant one, right? Because otherwise, it just doesn't make sense.
The footnote said that's like 10 per second.
Yeah.
Which is comparable to this.
Yeah.
So that, I'm still missing a factor of 10 to the 9 in here somewhere, but I'll think about that later.
Yeah. No, so I didn't expand it particularly here, but it comes down to how you go from this power use per cell.
Yeah.
And that requires an estimate of the typical genome length in those cells and how much of that genome is actually undergoing transcription. And to make that work, you come up, this is kind of the factor you get.
I understand your path, it's the path.
Right.
Mistake that the other people made.
No, I have not actually gone back to, I was so delighted that I was able to fix the problem. I haven't gone back to interrogate exactly where it went wrong for them. Okay, so just very briefly, all sorts of caveats to this, right? It's huge simplifications at play here. I'm talking about costed computation only, but there are other things going on to enable those computations in the first place. There's this vast ensemble of biochemical and biophysical processes that really one has to now start to do the bookkeeping for. And so this is just the beginning of the story. But let me, how much time do I have to?
Like 5-10 minutes.
5-10 minutes. Okay, no, that's okay. Okay. So let me do a worked example that is, still very, very simplistic, but just to illustrate how you can translate all of this into something that most of us would recognize as equivalent to something like a habitable zone calculation. So I'm going to talk about what I call phototrophic computation. So all I'm going to do here is say, let's think about the energy flow on the surface of a planet like the Earth from stellar photons. We know stellar photons are utilized by life in photosynthesis, and eventually photosynthesis doesn't necessarily do computation, but it produces the raw chemical energy that goes into computation. So I'm skipping some steps here, but just for the sake of the argument, and it sort of works because what I'm interested in here is that efficiency factor. So there's energy flowing through a system, but with what efficiency can I make use of that energy to perform computation? That's what I'm going to get at in this little example. So to understand this, and to come up with not an experimentally derived efficiency, but one based on thermodynamics. So this is like the best possible efficiency you could ever hope for in utilizing stellar photons. We have to worry about entropy. So some people go, oh no, entropy, I hate entropy, I don't understand it. Well, join the club. Nobody really does a good job with entropy. Part of the confusion that comes up when we think about this property of entropy is, the popular science way of talking about entropy is it's a measure of disorder. That's an okay way to talk about it, but it's not really accurate. So let me use the example of a pile of socks. Okay. So what entropy is really about, it's a measure of the number of microstates. Okay? And the more microstates you have, the more potential there is for stuff to appear disordered. That's why we make that jump to talking about disorder. So what do I mean by that? Well, okay, a pile of socks. This is kind of what my wardrobe looks like typically. Well, maybe not, that sock. A pile of disordered socks exists in a particular microstate, which is the particular arrangement of each of these socks to make that pile of socks. The macrostate is that there is a pile of socks of this rough size and this rough shape. So I could rearrange this, right, many, many different ways, just move these socks around in some random way, and I still have the same macro state. I still have a pile of disordered socks of roughly this size, but the individual placement of the socks is different. So each of those reconfigurations is a different microstate. What entropy is really a measure of is how many, or it's a measure of, it's actually a logarithm of, but we'll call it a measure of the number of those microstates. And so clearly a disordered pile of socks like this, and I'll use the word disordered deliberately there, is going to have lots and lots of potential microstates. If those socks were all carefully ordered and paired up, right, and lined up on my floor, there's only so many ways I can do that, far fewer ways than I can make this disordered pile. Therefore, my ordered socks have lower entropy. There are fewer microstates. There are fewer ways to arrange those socks in that more ordered fashion. So, entropy is also this measure of the amount of energy in a system that is not available to do anything useful with. And that relates to this idea of microstates, because one way to think about it is, I'm constantly reforming my pile of socks this way. A lot of energy just goes into those rearrangements without actually changing anything in the macro state. The higher the entropy, the more microstates, the more energy sort of gets mopped up into just moving between those different microstates and not actually doing anything to the macro state, not actually changing anything in the world. So in that context, entropy is also, in effect, this amount of energy that is not available to do anything useful with. This is just basically the second law of thermodynamics. Yeah, okay. I'll zip through some of this because of time. But what we're usually interested in is not what we can't do, but what we can do. And so there's a complement to entropy, and it's kind of an archaic term. I happen to really like it because it works well in this context, and it's called exergy. So exergy is the amount of energy that is available to do useful work in a system. Okay. And so therefore, the true limiting efficiency in any system is always going to be the exergy divided by the total energy. Right? This is like the unavoidable, inescapable limitation of efficiency, no matter what you're doing. So, okay, so in the interest of time, I'll pass through some of this rest of this fairly quickly, but I got interested in applying this in this context of computation to what happens when you try to utilize stellar photons in a very general form. And it turns out that you can calculate this exergy really neatly. There's a neat integral form. So if you have a Planck spectrum of electromagnetic radiation, the exergy of that radiation, so the amount of that, the energy in that black body radiation you can actually do useful work with, is this relatively simple expression, and it's in terms of the Boltzmann constant, but these two temperatures. One temperature is the temperature of the black body, or the temperature of the Planck spectrum. The other temperature is the system temperature. So that's your environmental temperature. It's the temperature in this room, or temperature on the surface of the planet. So you can quickly write the absolute efficiency is just that exergy divided by the total energy, which is the integral over the Planck spectrum. And it's, again, a simple, or relatively simple form. We've now even dropped the Boltzmann constant. Again, it's the relationship between those two temperatures that matters. Again, going through this kind of briefly, you can now take all of that and put it back into that simple idea for an upper limit to biological operations per second I had before, because there's an efficiency factor in there. So what I've done here is I've imagined a planet, and it's around a star, and I've worked out what the equilibrium temperature of that planet is at every different distance from the star. In this case, two stars. One is a solar mass star, one is a low mass M dwarf star. These two curves, the solar mass curve here and the M dwarf curve here. Sorry, the colors probably should have been switched around, but whatever. And so I've calculated the equilibrium surface temperature, but I've also computed the amount of stellar photons passing through the unit area on the surface of the planet, and the Landau limit for some hypothetical biological operation based on the environmental temperature. Put all of that together to give us a plot for biological operations per second per square meter on the surface of this hypothetical planet. So these two curves are kind of interesting because you look at this and first you go, oh, okay, there's something going on here that's interesting. Clearly, you're always going to get more out of a planet around a higher mass and hotter star in terms of absolute efficiency. The efficiency around the lower mass, cooler star is going to be systematically lower. The peak is where things get a little confusing because these vertical dotted lines are actually the radius of the star, the stellar photosphere. So it says that the absolute peak biological operations second, based on this measure of efficiency and how much power is flowing, is literally just outside the photosphere. Well, that doesn't make sense, right? How can that possibly be? Biology isn't going to happen there, and that's absolutely correct, right? And the simple reason is I've not incorporated anything about biology here. So what's missing? What's missing is some quantification of instantiation. When I talked about the three constraints, well, instantiation just means, you know, where should biological processes take place? What kind of conditions can they take place in, and so on. So in this example, it's the same plot, but what I've done is I've added a new factor, which is just this heuristic model of biological processes. I mean, I must have been very sleepy this day because I'll just use a Gaussian, right? So it's a Gaussian function centered at around 15 Celsius with a width of 15 Celsius. So it's meant to indicate the range of temperatures at which biological operations take place and how they drop off with variation from that temperature, which actually turns out to be pretty good. If you look at actual data on things like gene expression and translation as a function of temperature, it kind of does something like this. There's optimal temperatures for that. It's more complicated than just temperature, but there it is. So now my two curves for the low mass star and the high mass star shift way out here. Not surprisingly, they basically land where the traditional habitable zone would be because I built that into this. But it's no worse than saying there's got to be liquid water, or it's got to be between 0 and 100 degrees Celsius. It's just I'm being a bit more honest about my assumptions here in a sense. And these two peaks for biological operations, they look much more similar, but there is still a systematic difference. It's still systematically somewhat less efficient thermodynamically to be living around a low mass start. So, okay, I'm gonna just flick through the last slide and stop here. So this is kind of interesting. What we're gonna do with this next is start to put in some much more sophisticated models. So in this framework, what's interesting, what's nice is you can take, for example, simulations of planetary climate, something near and dear to my heart, where we have detailed maps of surface temperature. We have detailed maps of the amount of stellar photons absorbed at a given location that incorporates things like cloud cover and so on. We also have measures of aridity, or how much water there is at any given location. We can blend all of that in to that estimate, that upper limit of biological operations, and also to incorporate better biological models that relate things like transcriptional translation to temperature and to other properties in the environment. I don't know what the answer will look like. It's not going to be as simple as this. We're going to integrate this over planetary models and see what we get out at the end. Okay, in the interest of time, I will skip through Dyson structures because that's just really wacky. We need to drink some more wine before we talk about that. And I'll stop on this slide and just say, I hope I've given you a little bit of a taste. This is very new. It's sort of, some people have said to me, well, but you're trying to fix a problem that doesn't exist. We know how to do habitability and so on. And I just say, no, we don't. We don't know how to pull all those different threads together. This drills down as well. What I love about this is you can make use of fundamental physical principles, things like the Landau limit. That's inescapable. And we've not included that before in anything we've talked about in terms of biological operation. And it's dependent on the environment, it's dependent on temperature. So I would claim this is an inherently agnostic way of looking for life. It doesn't care how life is built. That's something you can explore as you're utilizing it. And it's not just going to be a sort of abstract thing, because we should be able to translate this into a map towards the same sort of biosignatures that we're already actively looking at, but interpreting those differently, interpreting those in terms of exactly what does that mean for the amount of computation taking place in a given environment. If we see a certain amount of oxygen or methane or nitrogen compounds in a planetary atmosphere, can we map that back to the core computational processes that really are what we mean when we talk about life, in my opinion. Okay, I will stop there. Thank you so much.
Hey, thanks.
Good, thanks. Yes, you talked about computation rates, but principal life can live on very different timescales, right? So it seems like you should normalize that to the amount of time you have to wait for your information to decay, right? Like your decay time, and that too could depend on temperature, right?
Yeah.
So how have you been thinking about that? Because what if your lifespan is just dramatically?
Yeah. So so this is great, right? Because it raises all these questions that we should deal with. So we've just started to think about that. What we're looking at for that as well is that in sort of theoretical computational study of computation as a phenomenon, people worry about coherence and decoherence in computation, decay times. When you create a bit, what's its lifetime? Right? And that depends on how you instantiate that bit and the environmental conditions and so on. We think there are some principles that people apply when they're studying information theory and computation, nothing to do with astrobiology or biology, but like the Landau limit, that there may be some principles that we can bring into play that are more fundamental.
But from a physics perspective, right, then it depends. It's going to depend on how you're encoding your information. Because just to take an overly simplistic example, if you're encoding your information in excited states,
yep,
then if you're sitting there and waiting it for just quantum decay, that might not depend too much on anything. But if you're sitting and waiting to be collisionally decayed, it's going to depend directly on the temperature. And so you have a very different,
absolutely. And I agree, and I don't have a particularly good answer at this point, except to say, that also raises the question of, so why is life as we know it built the way it is? Is there something about that decay rate for computation in DNA, RNA, cellular processes that is special or optimal for the conditions that we find ourselves in? So I can't really give you, it's a great question, and it's a great piece of this puzzle, because now it expands our conception of things. Maybe the only interesting computation happens in places like this, right, in the universe, and it happens at this rate, and it happens using this instantiation, but we don't know, right? So now with this, you kind of have to acknowledge that there are other possibilities. I mean, we do know that certain microbial species on Earth, there are microbes that living in the subsurface of the planet whose cell division time is like 1,000 years, right? It takes on average 1,000 years before they are going to divide because of their low nutrient environments. Clearly, computation is going to be very slow there. Is it pushing up against some limit, right? What do they do to repair DNA, right? DNA just sits there, stuff is going to happen, right? We don't know. But it all folds into this way of looking at things.
Related to Ruth's question, some of that idea about state lifetimes and temperature dependence is built into the definition of the Landauer limit, because it's for irreversible processes. And so implicitly it's some state change that won't go back. And of course, in infinite time, it will go back. But the reason it has a temperature dependence is you have to write harder if you want something to survive at 1,000 degrees long enough than to have it survive at 100 degrees. So it's
part you have to just have to define what infinite time is.
Yeah, so that's right. So in information theory, there's some discussion when they say irreversible, what do they really mean? But it's way at these long tails of time scales. So the sort of fundamental limit, it is built in. That's why you need more energy to do a bit at 1,000 Kelvin, 100 Kelvin, because you have to find a state change that lasts longer.
The body doesn't have to keep information for 1 million years, it just has to keep information for.
At the lateral limit, there's not I think, a big difference. But it's at least somewhat built into the information theory definition, what the state changes are, to think about how long, right? So,
and I do, to your point, I really like that you can think about this.
Natalie? I don't know.
You go ahead.
I'm kind of learning from that, so if you had a follow-up question that's related.
Go ahead.
Just thinking about heat sources and things, I mean, I know Joel's work on this, but radiogenic heating, how does that compare to the energy budget?
It's
Or stellar?
it's small. I don't remember what the actual figure is, but so I think usually people would translate it into a chemical energy budget. So radiogenic heating, right, maps into geophysical activity on a planet, which maps into the recycling and representation of raw chemical materials to the, let's say, to the near surface of the planet. I think that's in the, I want to say, tens of gigawatt level for the Earth. There are organisms, this is going off on a slight tangent, right? But there are organisms where the initial step in their metabolic process is radioactive decay, because it causes hydrolysis of water, it splits water into hydrogen and oxygen. These organisms then gobble up the hydrogen. There are organisms living in the deep subsurface of the planet where we actually know that to be true, that they rely on maybe thorium decay. And that triggers uranium and thorium. Yeah, it kicks into motion you split water, and bingo, you've now got consumable energy, but it originated as a atomic nucleus, as an unstable atomic nucleus. So it's a modest piece of the metabolic power budget for the Earth, for sure. Most is stellar photons. And actually, there's a caveat to that, which I had only recently really understood. So, and it has nothing to do with computation. So roughly 100 terawatts is kind of the power budget for Earth's biosphere, we think, based on stellar photon absorption and chemical energy. But if you include transpiration, what are trans something things here, but transpiration, so the movement of water through capillary systems in plant life, that adds something like 5 petawatts to the power budget. It's truly extraordinary. So in fact, the bulk of energy utilization by life on Earth for sort of non-computational purposes directly is in literally moving water on a daily basis from down here to up here, letting it evaporate. It's a huge, there's some really cool papers on this, just the sheer mass of water that's moved, but also that power budget. And I believe it is of the order of 5 petawatts, which is a more significant fraction of the whatever 75 petawatts that is actually absorbed at the surface of the planet. So it's going off on a tangent.
Are there any questions online?
I don't see any questions online.
Okay. Any other questions for David? We can continue discussions over wine if you'd like to stick around. We've got some food and wine and you can have informal conversations. So I think that would be lovely as well. So let's thank our speaker.